Today we will see about the presynaptic receptors. What are presynaptic receptors? Receptors located in or near axon terminals. On activation, they modify the function of terminal regions such as synthesis and release of neurotransmitters. So, what are the types of presynaptic receptor? There are two main classes of presynaptic receptors. One is heteroreceptors and one is autoreceptors. So, what are heteroreceptors? It is nothing but, for example, in this picture, we can see one sympathetic axon nerve terminal, and here there is one parasympathetic axon nerve terminal. So, this ACH released from this parasympathetic axon nerve terminal, if comes and binds to the receptors, muscarinic receptors present on noradrenaline synapse and causes inhibition of release of noradrenaline, then it is called as heteroreceptors. However, if this noradrenaline released from this axon nerve terminal comes and binds to the alpha receptors, alpha 2 receptors in the presynapse and inhibits its own release, then it is called as autoreceptors. So, simply to put, the neurotransmitter released from the nerve terminal, if it inhibits or increases the release of the same neurotransmitter, then it is called as autoreceptor. If the neurotransmitter or any other substance which is going to inhibit or increase the release of another substance, then they are called as heteroreceptors. So heteroreceptors are presynaptic receptors that respond to neurotransmitter, neuromodulators or neurohormones released from adjacent neurons or cells. So here are example of different neurotransmitters and molecules which act on these respective receptors can either inhibit or increase the release of noradrenaline. So this is the noradrenaline released from the axon, and these different substances which have the respective receptors in this nerve terminal, when they come and bind, so these all substances, this ACH, adenosine, PGE, they all will inhibit noradrenaline release. However, if adrenaline and angiotensin 2 comes and binds to beta 2 and AT1 receptors on this nerve terminal, then they will cause increase of noradrenaline release. So these are different substances. For example, adrenaline regulating noradrenaline release is an example of heteroreceptor. So here example is norepinephrine can influence release of ACH from parasympathetic neurons by acting on alpha 2A and alpha B and alpha 2C receptors. So when this noradrenaline goes and binds to this alpha 2 receptors present on the axon nerve terminal of parasympathetic neurons and regulate the release of ACH, then that is called as heteroreceptor. Similarly, when ACH comes and binds to M2, M4 receptors on adrenergic nerve terminal and regulates release of norepinephrine, then that is also called as heteroreceptors. Autoreceptors. Autoreceptors are receptors located on or close to axon terminals of the neuron through which the neuron's own transmitter can modify transmitter release and synthesis. Example is alpha 2 presynaptic receptor modulating norepinephrine release. So norepinephrine released from one axon will go and bind to the alpha 2 receptor on the same axon and it will modulate the release of norepinephrine. So this is an example of so again it's the same thing. Noradrenaline will bind on alpha 2A and 2C receptor on the same nerve terminal and inhibit noradrenaline release. Similarly, ACH released from parasympathetic neurons will bind to M2 and M4 on the same nerve terminal and inhibit ACH. So there is a combination of auto and heteroreceptor. This one example is histaminergic neurons. So in the histaminergic neurons, there will be release of histamine. So if this histamine goes and binds to H3 receptors of the same histaminergic neurons, it will act as H3 antagonist, and it will have all the central effects such as wakefulness, improve cognitive function, enhance memory, learning, attention, reduce food intake, like etc. And if this histamine goes and binds to some other axon nerve terminal, for example, adrenergic nerve terminal, H3 receptor present on the noradrenergic nerve terminal, then they will act as inverse agonist, and they will produce the similar central effects of H3 blocking activity. So once this receptor gets activated, how is the action produced? So they mainly act through G protein coupled system. So we would have learned about the different types of G protein couple system, GS, GQ, GI. So they will act through one of the G proteins, and they mostly act by regulating the second messenger, calcium. So they will either cause opening of the calcium channel, they will either cause inhibition of the calcium channel or opening of the potassium channel. So whenever calcium channel is inhibited, transmitter release is inhibited. Whenever the potassium channel is open, then transmitter release is inhibited as well. So sometimes they can directly act through ion channels as well. Important point to remember, when we say presynapse, we tend to think they mostly inhibit the release of the neurotransmitter, but they can increase the neurotransmitter release. Examples of presynaptic receptors are muscarinic receptors acted upon by ACH, alpha 2 receptors acted upon by noradrenaline and adrenaline, 5-HT1 receptors acted by serotonin, A1 receptors acted by adenosine, prostaglandin receptor EP acted by prostaglandin, H2 receptor acted by histamine, mu and delta receptor acted by endorphins, D2 receptor activated by dopamine, purinergic P2X and P2Y receptor activated by ATP, and cannabinoid receptor CB1 activated by endorphin. Summary: presynaptic receptors are receptors that are present at the presynapse of the axon nerve terminal. Their function is to regulate transmitter release and synthesis, thereby controlling the neuronal excitability. So if transmitter is released, there is neuronal excitability. Transmitter is not released, there is no neuronal excitability. So neuron modulation involves second messenger regulation of membrane ion channels such as calcium or potassium. And presynaptic receptors can both inhibit or increase transmitter release, the former being more important. They mostly inhibit, however, they can increase transmitter release as well. Example of autoreceptors are noradrenergic and cholinergic neurons acting on the same uh, presynaptic receptors. And examples of heteroreceptors are endogenous mediators such as GABA, prostaglandin, opioids, and other peptides, exerting control over noradrenergic or cholinergic autonomic transmitter release. Thank you. So the references of this lecture are from Rangan Dale and Goodman Gilman.